In today's video, we're going to be talking about how the body's immune system responds to invading pathogens. For this example, we're going to be using bacterial cells, and they're going to be invading through the epidermis into the dermis. So, to start off with, the innate immunity response distinguishes the body's own tissue cells and allows pathogens to be identified through pathogen-associated molecular patterns, also known as PAMPs. These PAMPs can be lipopolysaccharides, which are found in the envelope of gram-negative cells, or peptidoglycan, found in gram-positive cells, just to give a few examples. So, our antigens are indeed located on the cell membrane of our invading bacterial cells, and they are going to need to be identified by the body to produce an immune response. So, in order to recognize these antigens, our body has to have antibody receptors. A few of these receptors can be toll-like receptors or nod-like receptors. Nod-like receptors activate transcription genes that promote defenses such as autophagy. Toll-like receptors, on the other hand, allow for recognition of potential pathogens, and cells stimulated by toll-like receptors secrete chemokines, which allow it to recruit other cells of the immune system, and cytokines, which promote different aspects of the immune response. Our beta lymphocyte right here typically floats through the body or circulates through the body through secondary lymphoid organs until activated by a specific antigen. The antigen in this case was indeed found on our bacterial cell membrane, and it was picked up by our toll-like receptor right here. Now that the beta lymphocyte has been activated, it is going to result in memory cells and plasma cells. These plasma cells produce about 2,000 antibodies per second, and these antibodies are going to respond to the antigen presented on the bacterial cell envelope. So the antibodies are going to bind to the antigen and basically mark the cell for degradation. However, it is important to note that the combination of antibodies with antigens does not itself cause destruction of the antigen or the pathogenic organism that contains these antigens. Antibodies rather serve to identify the targets for immunological attack and to activate non-specific immune processes that destroy the invader. The ability of antibodies to stimulate phagocytosis is termed opsonization. Now that our invading pathogens have been marked by the antibodies, a phagocytic cell can indeed identify them and ingest to them to break them down through the use of lysosomal enzymes. The phagocytic cell can also excrete these lysosomal enzymes into the extracellular matrix to break down the pathogens instead of ingesting them. This is a good segue to discuss the complement proteins. Complement proteins are designated C1 through C9, and these proteins are present in an inactive state within the plasma and other bodily fluids and become activated by the attachment of antibodies to antigens. The function of complement proteins can be subdivided into three groups: recognition, which is performed by complement protein 1 or C1, activation, which is done through C4, C2, and C3, and attack, which is C5 through C9. The classic pathway of complement-dependent cytotoxicity is initiated by the binding of immunoglobulin G antibodies to their cell surface receptors. So let's say that step 1 for this complement protein pathway is going to be found here. If we zoomed in at this point right here, give me 1 second to draw it, sorry, we're going to find that the cell membrane has antibodies attached to it. And these antibodies will indeed have the complement protein bound. Let's change the color, I'm sorry. Here. So this is complement protein 4, and at this point, complement protein 1 has already been activated when the antibody bound to the cell body. Okay? So this hydrolyzes or catalyzes the hydrolysis of C4 into two fragments. So it's going to split here and it's going to detach. So C4 is going to split into two pieces and C4A and B will be released. C4B will then bind to the cell membrane and becomes fixed and becomes an active enzyme. C4A will be released and will act as a chemotaxis mechanism. Through an intermediate step involving the splitting of C2, C3 is also cleaved into C3A and B. The C3B converts C5 into C5A and C5B. So to summarize that in an equation, C2 hydrolyzes 3 into C3A and C3B. C3B will then convert C5 into C5A and C5B. C3A and C5A will act as chemokines and attract additional macrophages and neutrophils, monocytes, to the site of infection. C5b, in addition to C6, C7, C8, and C9, will embed themselves into the membrane of the cell. So C5b, C6, C7, C8, and C9 are now embedded into the membrane and form the membrane attack complex. This attack complex is a large pore that can kill the bacterial cell through osmotic influx of water. As mentioned earlier, C3A and C5A act as chemokines, but they can also stimulate mast cells. Once the mast cell has been stimulated, they're going to be releasing histamine. And this histamine will basically cause dilation of the capillaries and blood vessels and increase the permeability of the capillaries. In simpler terms, the increased dilation will allow an easier opening for more phagocytic cells to travel through the capillary and to the site of infection, which will basically allow for more immune response and a quicker response to the invading pathogens.